Welcome back. So we are on part 2 of our nucleic acid metabolism. So, nucleic acid biochemistry. So this is a double helical DNA strand. Double helix. One strand is a helix. Another strand is a helix. So we call it what? Double helix. So let's see it. So we have one strand is here. It's a helix. We call it a helix. Another strand is here. We call it a helix. So one helix plus another one helix. This one is also a helix. We call it what? Double helix. So double helix. Okay, so let's continue. So when we say denaturation means that it's the same as unwinding. Denaturation is the same as unwinding. 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 Unwinding, meaning that separation of the one strand from another. So one strand of the DNA separates itself from the other strand. We call it unwinding. So you can see this one unwinds. So the bonds between the guanine, cytosine, adenine, thymine breaks. The adenine bond breaks. So we have what the strand has. So we call it denaturation. In the end, we have a partial denaturation. This is a partially denatured DNA, and this is a fully denatured DNA. The strands it's like two married couples that have divorced, or two Siamese twins that have been surgically separated. So separated strands of DNA in random coils. So randomly they are coiling. So we have, if the DNA structure, if this separated structure, if their pastor counsels them that no, divorce is not good, come together again. Bible says the two shall become one. Then they will swallow their ego or their pride and bond again. So the nitrogenous bases will bond again to produce this partially renatured, partially annealed structure. So we have this over here, association of strands by base pairing. We will have this one. Then they will say, the woman will say, me, I don't love him, I love him, I don't love him, I love him. Then small, the man will coerce them, and the pastor will coerce them, and later they will join again, and they will lie on their bed and they are naked, and they will feel no shame. So we call this process of annealing. Annealing means that these separated strands coming together to bond together to get something like this is what we call annealing. For instance, if Sibdo anneals to Hannah, we say they are married. Annealing. Or they can anneal like this. Annealing. They are bonded. So they form a double helical structure. It's called annealing. But if Hannah says, I'm packing my things and go. So Anna will pack her things and go. Sibdo will also pack his things and go. They have what? Denatured or divorce. Denatured. Denatured. But here, if they've come together, we call it annealing. Annealing. They have annealed. They have combined. They have married. So annealing. Cool. Okay, so cellular damage. What are the factors that can damage a DNA? One, there can be UV light exposure will damage the DNA. That's why people, some of the time, if you are in Australia or New Zealand where the sun is too high, and also Africa, you end up getting skin cancer. The white people come here, they get skin cancer. Ionizing radiation, if you are working at the X-ray unit, gamma rays, beta rays, gamma rays, if you work at those places for too long, you end up getting high doses of ionizing radiation, which will later transform to genetic mutations or DNA damage. Genetic mutations. Mutation means what? DNA has been injured. There is what? A change in DNA structure, and most of the time the change or the mutation is in what? The nucleotide sequence or nucleotide. And if it is a single nucleotide, we call it single nucleotide mutation. Single nucleotide mutation. Nucleotide mutation. So mutation is any abnormal change in DNA structure, and most of the change has to do with what? Most of the change has to do with the nucleotide sequence, not the ribose or the phosphate group. So we have ionizing radiation, X-rays, gamma rays, delta rays, and the rest. Chemical exposure. Some chemicals are called alkylating agents. Alkylating agents. Alkylating agents. They bond to the DNA structure and cause mutation. Alkylating. Let me bring a new one. We have alkylating agents. Alkylating agents. There are some chemicals that you inhale. There are some drugs that you take and other things which produces what? Mutations. These ones are called alkylating agents. Any chemical agent that causes what? Cancer. They call alkylating agents. So we have alkylating agents, that chemical exposure, replication, another alkylating agent, another chemical, cyanide. Cyanide, those that they work at the mining company. If your father and your mother are doing galamsey, they have to be careful. They can end up causing mutation to their genes. Mutation, single nucleotide for polymorphism, mutation to their genes, and they will end up getting what? Cancer. And the cancer is heritable if it is in what? If it is in the germ cell. The gamete and the what? The gamete. If it is in the male gamete, the testes and the ovaries, it will be translated to the rest of it will be translated to what? The next generation. So cancer, breast cancer and others, some of them is not only genetical, it's an inherited genetic defect. Some of them is just through inheritance, but some of them is acquired. So we get the inheritance one, and another hit will end up getting what? A homozygous dominant or homozygous recessive for BR or BRCA1, BRCA2 genes for breast cancer and the rest. So we have what? Cell cycle checkpoints. These are checkpoints in the cell cycle that mediate repair of what? Damaged DNA. So as these ones are damaging, God has put cell cycle checkpoints. God has activated them to repair the DNA. Transcription program activation. We also have what? We have genes like P53, which is an apoptotic gene. P53. It is the main gene that repairs DNA mutations in a cell. It repairs. If it repairs and is not able to repair, it will let the cell undergo something we call apoptosis. Apoptosis is called apoptosis or what? Programmed cell death. Apoptosis is also called programmed cell death. Apoptosis so it's triggered by the p53 gene, and this is what a checkpoint in the DNA or in the body that repairs what damaged DNA. It's a gene that produces protein that repairs damaged DNA, and the protein it produces is called, we have the BAX protein, or it enhances the BAX gene to produce BAX protein, BAX gene for apoptosis. Okay, so these are the various DNA repairs: direct, reversible, base excision repair, nucleotide excision repair, mismatch repair, double-stranded DNA repair, break repair, homologous recombination, non-homologous. Please, this one is too early. I will teach you some of these things later. You are young to know these things. At other functions of nucleotide. So nucleotides carry chemical energy in cells. They also serve as signals like cyclic AMP, regulatory molecules, and components of many enzyme cofactors. So you can see they store chemical energy. That's why we have the ATP, GTP, CTP, and what? AMP. We have the ATP. Let's look at the chemical energies. So when we say the energies of the cell, or what the cell uses, the main energies of the body are what? Let's start. 1. GTP. GTP. ATP. CTP. UTP. TTP. This is the energy the cell uses. So we have adenosine, guanosine triphosphate, adenosine triphosphate, cytidine triphosphate, uridine triphosphate. Then you have thymidine triphosphate. Because it's triphosphate, when we break one phosphate, it releases an energy. When we break two phosphate, it releases more energy. Then we get AMP. And the AMP or GMP or CMP or whatever is the low energy charge. But if it is GTP, ATP, that is high energy charge in the cell. High energy. So if you have a lot of this GTP, ATP, and the rest, if it is triphosphate in your body, then it means you are satisfied. You have a lot of energy in your body. But if they change to AMP, if they change to GMP, if they change to CMP, if they change to TMP, monophosphate, all of them are monophosphate, and if they change to UMP, it means you are very, very hungry, or you are what? Deprived of energy. Low energy state. It means that you need to eat. You don't have energy. Your cells do not have energy. Your enzymes do not have energy. So we also have the cyclic AMP, that's the cyclic structure formed from the phosphate group and the ribose sugar. They form a cyclic structure called cyclic AMP or CAMP. And this one mediates signal transaction. You will see it later as you progress in your career. We also have regulatory molecules. So these nucleotides form regulatory molecules. These nucleotides form part of what? Enzyme cofactors. If you can remember, I taught you NAD. This is what we call nicotinamide adenine. There's adenine there, dinucleotide. Nicotinamide adenine dinucleotide. That's reduced nicotinamide adenine dinucleotide. And this is the oxidized nicotinamide adenine dinucleotide. This is the oxidized NAD. We also have what? So this one, we also have what? Nicotinamide adenine dinucleotide phosphate, the reduced one, and we have the oxidized one, nicotinamide adenine. This is a coenzyme, and so it's there is what? Adenine there. So the adenine there is contributed by what? A nucleotide. So these are some of the functions of nucleotides. So enzyme cofactors. So let's go. The next. So let's see. I was teaching you. So when we break one of these phosphates, it releases a big energy here. So energy released for cell metabolism. This is it. So we get what? We end up getting what? Adenosine diphosphate. Nucleotides also serve as signals. They serve as signals. So, like I was saying, cyclic AMP. If you have extracellular signal, it gets onto a receptor, and the receptor uses what? Cyclic AMP or cyclase. We have nucleotide substrate producing what? Cytidine CNT, which is what? A nuclear response element, going back to the nucleus to affect genes that will produce cellular response. These ones are complex. Later we will be doing signal transduction as you follow through with your career. So nucleotides serve as regulatory molecules. So nucleotides are part of RNA and DNA. Their degradation produces nucleoside bases. That's when you remove the phosphate. And they are also catabolized, or they are salvaged, or we mobilize them to produce nucleotides. But if we break them down, we produce nucleoside that's without the phosphate group. Okay, so we did something about enzymes. So nucleotides serve as cofactors, like what I showed you, the NAD and the FAD. We also have FAD. FAD, anytime you hear we have FAD2, FAD, so we have FAD plus or FADH. The A here is what? Adenine. This is what? Flavin adenosine, reduced flavin adenosine dinucleotide. This is oxidized flavin adenosine dinucleotide. Okay. So this is the cofactor, inactive protein. There is an adenine or guanine or cytidine inside the cofactor. So produces, when the cofactor binds onto the enzyme, it becomes active before the substrate and others can also bind. So let's look at application of gene therapy in cancer. Gene therapy, that's when we replace the gene. It forms part of cancers, inherited disorders, infectious diseases, immune system disorders, vaccination, so HIV and others. If we change the gene that is causing the problem, we will remedy the situation. So the principle of gene therapy means that if a gene is causing a problem, you cause a gene that does not have that problem, put it in a cell, produce more of the protein. For instance, if the person is not able to produce insulin for diabetes, to be able to control sugar in the blood of a diabetic patient, then you take insulin gene from a human being, put it in a yeast cell or in a bacteria, and it will produce more of what? The insulin or therapeutic protein, so that we can go to the pharmacy shop and go and buy and inject ourselves. So if somebody has diabetes, he goes to the pharmacy shop to go and buy insulin to be injecting himself. That's those who have the type 1 diabetes mellitus. So you put the gene of interest from the human being, you put it in a cell culture or in a bacteria, culture it, and let the gene produce more proteins, harvest it, and sell in the pharmaceutical industry. So people are making money, a lot of money, from molecular biology or gene therapy. You take a human gene, normal person who is producing insulin, take the gene, put it in a bacteria, then the bacterial cell will be producing more of the therapeutic protein. You harvest it and go and sell. This is what we call what? Genetic engineering, or biotechnology, or genetic recombinant DNA technology. Application of gene therapy in cancer. In gene transfer, researchers introduce a foreign gene directly into cancer cells or into surrounding tissue. The goal is that the newly inserted gene will cause the cancer cells to die or prevent cancer cells and surrounding tissues from funneling blood to tumors or depriving them of nutrients they need for survival. So let's see. So let's go to the next. This is so complex. So if you have inactive virus, we'll be doing this in immunology. Inactive virus. This is a T cell. So this is the virus. The virus will bind to T cell because T cells mediate cell-mediated immune response. They are cells that fight organisms. So it will bind to it will enter the T cell. When it enters the T cell will also produce what? Chimeric antigen receptor, these yellow ones. And the yellow ones will bond to what? The cancer cells. The cancer cells, and attack them and kill them. So we have the chimeric antigen receptor, that's a CAR T cell, latches onto and attacks cancer cells. So we use an inactive virus. It's like a vaccine we are developing. We use an inactive virus, put it in a normal T cell of a normal immunocompetent human being, and the virus will enter the T cell. The T cell will produce chimeric antigen receptor. That's, if you say chimeric antigen receptor, an antigen receptor that resembles that of the virus, then it will now fight cancer cells. Let's look at the next slide. So application of gene therapy. With gene therapy, the treatment or elimination of inherited diseases or physical conditions due to these mutations could become a reality. Gene therapy involves the manipulation of genes to fight or prevent diseases. Simply, it introduces a good gene into a person who has a disease caused by a bad gene. So if you have a cancer, breast cancer, and it's as a result of a bad gene, and the gene that mediates breast cancer, we call them the BRCA1. This is breast cancer in females, breast and ovarian cancer, breast cancer in females. And this is BRCA1. Then we have BRCA2, or BRCA2. It's called BRCA2. This is breast cancer. This causes breast cancer. The first one is breast cancer in females, or breast and ovarian cancer in females. This one is breast cancer in males. Breast cancer. We think it's only female that get cancer. Males also get cancer. Go to Thermal Teaching Hospital, you see breast cancer in. Go to Konfanochi too and you see in females. In males. Breast cancer in males. So males are also getting breast cancer. It will be there. Okay. So BRCA1, BRCA2, BRCA1, BRCA. So application of gene therapy, we have adeno vector. We put a correct gene inside. We'll put it inside the body, and to go. This is a vector. A vector is just a carrier. So adeno vector will carry a correct gene. Go and put it in somebody who is suffering from cancer or something. It will go and incorporate it into the person's DNA. So correct gene will go and insert into the DNA. then the defective gene. So we have the correct gene, that's the blue. It will go and correct the defective gene. So you can also gene therapy for sickle cell disease, collection of blood forming stem cells. Stem cells are what? Baby cells that form the normal cells. So for red blood cells, we have the stem cells are called reticulocytes. Reticulocytes, they are the stem cells for RBCs. Stem cells for our reticulocytes. So people with leukemia and others, you see a lot of reticulocytes. So stem cells corrected by beta globin gene transfer from viral vectors. So when we correct the stem cells, then we put it back into the human being. So gene therapy, you can go and read the rest. Go and read this. The rest I've explained. Autoimmune diseases and the rest. Gene application of gene therapy. These are vaccination. I will teach this one in immunology. But let's look at this. Let's see. Gene therapy may be used to intervene in the spread of infectious agents at the extracellular level. This could be achieved by sustained expression in vivo of secreted inhibitory protein or by stimulation of a specific immune response. So we have those. The application of gene therapy in autoimmune diseases represents a novel use of this technology. When you say autoimmune disease, the body producing antibodies or statutory T lymphocytes to fight against its own cells. So we can example is in the rheumatoid arthritis and the rest. He said the body will produce its own. The body will produce antibodies, statutory T lymphocytes to go and fight its own body cells. So, for instance, if somebody has a transplant surgery, we realize that the body, somebody you are there and they give you another person's kidney, your body will start fighting that kidney because it doesn't know it. That kidney belongs to somebody else. They are stealing it for you, even though you bought it. By nature, God gave you your own kidney, so why should you go and get another person's kidney? So such people, they take in a lot of immunosuppressant drugs to stabilize their body. They've taken a lot of immunosuppressant drugs so that the body, the antibodies and the statutory T lymphocytes they produce will not go and attack the organ that was donated to them or they bought or was transplanted into them. The goal of gene therapy in the treatment of autoimmune disease is to restore immune homeostasis by countering pro-inflammatory effects of CD4 plus T cells in lesions of autoimmunity. Later we will do these things in immunology. Thank you very much, class. May God bless you.